Hello, everyone. Welcome to this new seminary of SEM sessions. So today we have Alex Baumel, that's been invited by Erol, and enjoy today's seminar, and I'll let you introduce the speaker. Thank you.
Thank you. It's a pleasure to present you my colleague Alex Baumel, who is teacher at Aix-Marseille University and researcher at IMBE, Institute of Mediterranean Biodiversity and Ecology. He's a field ecologist and specialized on genetics, and he's working on mainly on conservation of Mediterranean flora, and he uses genetics for solving some problem like taxonomy, like population genetics and evolutionary potential and problem of conservation.
Okay,
Alex.
merci. So thank you for the invitation. So my talk is on the difficult problem of the mismatch between the species, which is a taxon of species rank, so a taxonomical problem, and the species as the object of evolutionary biology, the unit of biodiversity, and from outside, species is the main object of biodiversity. So attends. C'est bon, c'est bon, donc je dis que ça. Okay, so with my colleague Didier Aurel, we published last year a paper in Naturae named Uncertain et taxon, so the uncertain species and the fuzzy taxa. And Didier is working on marine animal, and he has the same issue of this mismatch. So species are conceptual objects that are very useful to identify, to observe, to study biodiversity. But in reality, organisms are not species, they are population. They are population of organisms interacting with other organisms, with their environment. And the fact is that organisms and population of organisms are challenging their classification as species. And when they do that, this creates a great diversity and the creativity of name in the scientific community. So for Darwin, for example, this is a dubious species or the doubtful species, which is not a negative argument. It's just an argument to explain the transformation from the population to the species, to the variety, the subspecies. And Darwin also introduces the word of incipient species, so new species and an ongoing process of speciation. And Queno used also this word in a paper on Sepia officinalis, and as Sepia officinalis, une espèce en voie de dissociation. Well, 1 century ago, Lotzi proposed the word of Syngameon, and he was working on Saxifraga and Betula, which are genus of plant with a lot of hybrids. And a Syngameon is like a community of reproduction above the species level. This is an interbreeding network of distinct species. So what does it mean? Is when populations are challenging their classification as species, most of the time, this is because they exchange genes during speciation and also after speciation. So I think that everybody agree on the fact that gene flow can occur during speciation, but it's more or less difficult to accept that it can occur also after speciation. And well, the evidence of that one of the most striking evidence of gene flow after speciation is cytoplasmic capture. So Aviz, 22 years ago, published a book with a list of examples of cytoplasmic capture in animal and plant. So capture of the mitochondrial genome or capture of the plastidial genome. And now we have just a lot of more examples of cytoplasmic capture. In some cases, it's it's not possible to use the cytoplasmic genome to decipher the relationship between species because there is so much cytoplasmic capture that it's not useful as information for phylogenetic or taxonomy. And the process behind this, is what we call genetic introgression. So this is the incoming of the gene of a part of one genetic material in the genome of a second lineage of species. And if there is some disequilibrium between effective size of the population, which are hybridizing, this can lead to what we, at the end, we call cytoplasmic capture. So this requires that despite possible lower fitness of hybrid, genetic introgression requires a lot of generation of genetic exchange. And the second evidence is, genetic admixture, that we commonly see this type of figure within species. For us or species, for example. But here, this is a paper published last year in the North White American oak Salgamion, and with genomic data, and 13% of the genotype were admixed. So either first generation, backcross, or even more deep admixture. And most of them was with the widespread Quercus macrocarpa, which is the main dominant species in this lineage. And an interesting point is that the unique predictor of admixture was the distance to the distribution margin. So the distance to the frontiers of the distribution. And this is often seen in Sangamion, the correlation between ecological limit and the increase of admixture. Another evidence is adaptive introgression. Here, this is a well-known illustration of that in Lepus americanus, so the snowshoes hare, which is so a species that is molting in autumn, so it changes of color to become white during winter, and so it's a North American species, but it occurs about the same example in Europe, and some population on the northwest, so near the Pacific coast, at lower are living at lower elevation with less snow in winter, and they stay blonde. They keep their blonde color during winter. They do not molt, and this winter blonde phenotype are here. And when the analysis is done at the genomic level, the winter blonde population are clustered with their species. But when the analysis is done at the level of the agouti gene, which is involved in the process of coloration, then the winter blonde population are associated to another species the black-tailed Florian. So it means that during the adaptation of this population to a new environment, relatively to the environment of the rest of the species, the allelic, the genetic gene variant, the allele, involved in the adaptation was not coming from a mutation de novo or from variation already existing in the population, but from another species. And so genetic interrogation is considered as a facilitator or facilitation of local adaptation. So the frequency or the importance of this kind of process is not evident, but it is probably one of the main processes that explains that sometimes we have some difficulties with identification and species delimitation. And so I will present two examples from my research on that. The first one is on an arrow endemic of Dauphiné, which is a Saxifraga delphinensis. So it was discovered and described by the I don't know the word in English, le curé de Villard-de-Lans, Raveau, was churchman, and was a botanist in 1885, and a very good botanist. And well, he described this plant and he said it's very similar to some Saxifraga from Pyrenees, because of the airiness, but also the white flower. So it is lying from Grand Vémont, Pas de la Ville, up to the last summit of the Dévoluy on the east, Baronnie, and Esaü. Esaü is the westernmost point of the Alps. It's amazing to see that when you go on a map, at the end of the Alps, you will find some Saxifraga plant, and it's at low elevation, so maybe like 700 meters. And so we work on this plant with several botanists from Conservatoire Botanique Alpin, from colleagues also from Grenoble, and from Jura, Massif Central, etc. And so in this area, Saxifraga delphinensis is often in sympatry or very close to other species, Exarata, which is widespread in the Alps, Moscata, which is widespread in the Alps and Pyrenees. And in the past, the two species, delphinensis and Moscata, were subspecies of Exarata, but with no studies. That was just put as subspecies just for commodity. Yes. They are rather easy to differentiate. It's for botanists, they are good species, not fuzzy species. And on the basis on the compactness of the cushions, airiness, the size of the leaves, the form of the leaves. And when we integrate this data in a multivariate analysis based on the only vegetative character, so you can identify the plant during all the year, it's very clear that delphinensis and Moscata are well separated. Exarata also form a cluster, but as you can see, some plant from Exarata can be confounded with Moscata or with delphinensis. For example, at the summit of Mont Ventoux, there some Exarata have the very compact cushion of delphinensis, but they are Exarata. It's rather clear when we look at the leaf. And so with RADSEC data, so a representation of genome, so it's like genomic data, multilocus data. It's clear that there is some gene flow between the species. Most of the unmixed genotype are in Exarata, and some have free ancestry. So it's interesting. And but we found also some deficiencies with being admixed. And most of the admixture was observed in on the plateau, the Pic de Bure, which is a big area at almost 3,000 meters of elevation. It's dominated by cushion plant like this Saxifraga, and the free species are very frequent, very abundant, and in sympathy. And on this place, always on the summit of Vercors, we observe some intermediate phenotype. When we look at the main barcode of plants, so the EKS, deficiencies as a cluster, but it is also observed with the one not frequent but widespread haplotype in Exarata, and it has also I found also some individual of deficiencies with the haplotype 3, which is very common in Muscat. So with different observation, we observe that deficiencies. So it's not a monophyletic lineage. We did some phylogeny with the RADSEC, and it's clearly not monophyletic, it's rather polyphyletic. We observe we show some evidence of ongoing gene flow, but also past gene flow with the ABABA test of interrogation. But it has a distinctive morphology, and it's forming a fuzzy genetic cluster. So I cannot define the good species. I cannot give a good definition of species for this plant, but I think this is a good species. And maybe the minimal definition of James Maley on designing the species as a genomic cluster could be worked here. But so I need to pick the good definition. It's rather a bit weird to have to choose the definition for a taxa. So the second example is on a smaller area. It's in the National Park of Calanque. So the National Park of Calanque is at the south of Marseille. I think you know the place of amazing landscape, very wild landscape. We are very lucky to have two archipelago of Iceland. Two different one in terms of ecology, in terms of also of history. This one was very close to the coastline during the last glaciation. This one was far from the coastline during the last glaciation. And this is a place of big originality for fauna and flora, but also a place of great vulnerability. First, there was a lot of, during all the history of Marseille, a lot of activity in this. So, for example, the military buildings are very frequent on the freehold and in this part of the coastline. Several battles occurred, and with all the pollution associated to the bomb and etc. There is also many places of historical place of exploitation of the sand and gravels, and also several sewage. The main one is Cortu, but there is a lot of smaller in the city. And during the history of Marseille, the industry of metal was transferred from the center to the south, And during the 19th century, up to, well, the beginning of the last century, there was a lot of pollution by heavy metal. So it's a nightmare. And the wind spread the pollution on all the western part of the Calanque and also in the sea. And we observe both on the soil and in the sea some evidence of pollution. And this limit, for example, are just it's difficult to design a limit for pollution because we don't know what are the concentration that could be dangerous. But this is the limit that could be dangerous for human. So I don't know if for plants or for other animals, but there was also in the last 20 years, 30 years maybe, a big increase of the population of the seagull in the Iceland with eutrophication due to the waste in the sea. Okay, so we have in the world 300 Teucrium. So Teucrium genus are the germander. We are in the Lamiaceae family. So the family of mint, etc. Rosmarinus, Salvia, little thyme. In Bougeron, we have 12, 13 taxa of germander, and in the National Park of Calanque, 7 taxa. So I will focus on the three taxa. Polium subspecies Polium, which is with white flower, white tomentum. So the tomentum is the hairiness of the inflorescence of the capitula. It's very important for the plant to protect the flower. And white flower, white tomentum, a small open shrub, difficult to maybe more hemicryptophic. And it is a tetraploid with 52 chromosomes. The golden germander, Teucrium aureum, yellow flower, yellow tomentum, a cushion, living on rocks, mainly on rocks, more very adapted to the stress, wind stress, temperature stress of cold and warm temperature on the summit of the small mountain around Marseille. It has a cushion and it is a diploid with 26 chromosomes. And the Polium subspecies purpurulescens, which is the only protected germander in the area, with magenta flower, white tomentum. This is a small dense shrub. Could be like this sometimes, for this 8, and this is a diploid. This plant has been extensively studied by Suzette Puech from the Botanical Institute of Montpellier. She did her thesis on this plant, and she described purpurulescence. Before Puech, purpurulescence was a variety of Polium, and she found that it was a diploid and decided on the basis to put this plant at the subspecies rank. Not the species, I don't know why, but at the subspecies rank. And she said, okay, it is present on the Frioul Iceland and Montredon. Montredon is just at the south of Marseille. So Montredon is this place. Okay, so keep in mind this phenotype, and well, we observe several other forms in the Calanque. So such this one, so yellow flower and white tomentum, or this one, white flower and yellow-green tomentum, or this one, so it's amazing, yellow flower, white tomentum, but you see that the gynaecea, the androcea, and the epiderm below the hair are mostly red, so purple, by accumulation of the anthocyan pigment, which causes the color magenta. And sometimes bicolor plant, well known by Erol. I think you spent a lot of time during when you was young Marseille on this plant, and so red and yellow. But if you see the gynaecea and the androcea is red. Okay, So we did a collection of pictures. We organized this with students 2 years ago, and as you can see, there are 3 species. It's very clear. They are present here and on the top. Now there is a huge variation, and it's unique in the world. So germander are very difficult plants and with very variable, but such variation on a small place is unique to my knowledge. So we count the phenotype. So this is the expected phenotype with 3 variables: the first color, the second color, if any, and the color of the tomato. So color of the petal and tomato. So this is magenta, magenta, white, the yellow, 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 and the white, white, white. And so the expected phenotype are, well, seen in majority. So on 712 phenotype, we observe 63% corresponding to the expected one, and the unexpected one correspond to 37%. So it's a lot. And most of this unexpected phenotype were the yellow, yellow, white tomato with represent 16%, so 118 plants. Of course, this is a very simple way to design the phenotype. There is a huge variation between these, but it's useful to present you the context, in fact, of the variation. So the magenta color is decreasing from the triole up to the cat module, the last point, with magenta, purple pigment are here. The circle corresponds to the expected phenotype, the triangle to the unexpected one, and most of the diversity of phenotype is observed here. So this is a Monroe. So QH describe fluorescence from triole and from here. But on all the coastline here, there is a lot of recombination. It is less frequent in this part. Okay, well, as I say, recombination, I don't know what. So we estimate a genome size with flow cytometry. Flow cytometry is working well on germander to differentiate diploid and tetraploids, even on dried material. So at least to differentiate diploid and tetraploid. So it's useful. And so on the Iceland and the coastline, we found only diploid. On the plateau of Calanque, we found some stand mixed with diploid and tetraploid, corresponding to hybridization between pollium and aureum. But we do not observe triploid, so do not ask. I don't know where they are. Maybe this hybridization without triploid. And so this is an interesting place. This is a site which corresponds to the old coastline. So we are on sand under a pine forest that was previous was a coastline with a lot of sand that was exploited. So many destroyed, with a lot of rare species from the coast, from the littoral, as well as some insects from the littoral also. And here most of the plants are tetraploids with all the colors. So we observe also tetraploids with the magenta color. So this is probably another case of hybridization. And so we look on the elevation. This is the Marseille vert. So I just exacerbate a bit the relief. So we see that the magenta color is decreasing with elevation, and the unexpected phenotype is increasing up to this part and then decrease. Okay, so we are currently collecting genetic data. The genome of the two Aureum and purpurесcens is under sequencing in Toulouse, GenoTool platform. And this is targeted sequencing data. And genotype from Frioul are well isolated. On the morose, morodon, so this is purpurесcens. The morose morodon is here, close to the Riou Iceland, which is the second archipelago of Marseille, at the south of Calanque. And Saint-Baume is a mountain rather far from the study area that is used to represent the Teucrium aureum, the golden jamander. So if I add the phenotype, this is like this, the yellow, the golden jamander, the place of the maximum of phenotype diversity. And on the Frioul, most of the plants are magenta white, but some are a bit different. But with the yellow pigment is not present in. About taxonomy, PH described purpurесcens like this, but then the knowledge change, and the botanist, so Erol, Daniel Pavon, the colleague of National Park, call all these plants purpurесcens. And the reason of that, I think, that was difficult to, and even in Flora Gallica now, all of this is purpurесcens. And the reason of that is that this plant is protected also. So that was one reason. Okay, so we did some phylogeny. So with the morose, the Riou, the Frioul, the golden jamander, Polium, so species Polium, the tetraploid, and Montanum, because Montanum is known to hybridize with Aureum. So, and we know that some hybrid, but, and so we put Montanum apart from the outgroup. The outgroup are other species. And with the Ababates, we look at introgression and we found the contribution of Polium to Aureum. Okay, so we do not use the hybrid on this, the evident hybrid in this analysis, but we found kind of a link between both, and also a contribution of Aureum to Riou and to morose, and well, that explains the yellow pigment. So we also simulate hybrids, so first generation hybrid only between the Frioul and the golden jamander, and we found them in this place in the analysis. So it means that since the hybridization, there was a genetic drift, genetic evolution, I don't know, selection, adaptation, probably. Well, j'ai presque fini. So, as you see, there is clearly some uncertain boundaries between these species of Teucrium in the National Park of Calanque. Separately, they are easy to identify, but if you start to looking and working on these plants, it's not the same game. And well, we are waiting a lot of result for the next year, and well, we'll do some communication about that. So the last word are uncertain species of Uzi taxa are more than a taxonomic issue. They are, for me, an amazing illustration of evolution, of ongoing evolution. And of course, taxonomic issue is the problem, but we should not overlook this plant, both for science, but also for conservation. And there is a lot of to understand this on the way. I think this is mostly a human problem. The species is mostly a human problem. to find the good, the best strategy to consider this complicated case of taxonomy. And so I have a lot of people to science for Saxifraga study and also Teucrium. So Mathieu Charrier, Luc Garot, Sylvain Dulac, Cédric Dantan, who are botanists and give material and hints. Karim Fremel, étudiant de Marseille, Jeanne Smica, qui a fait sa thèse à Grenoble. Sorry. Christina Roque and Sébastien Lavel, also from Grenoble, and Christina Roque is now in Barcelona. And for the Teucrium, we are supported by the Amidex Foundation, and this is the IAM project, and we are working on Diamander. I'll show you some results, and also on Gorgonian animal, marine animal, so the genus Unicella, which is with also a trio of species that are under introgation with Didier Aurel. And so Daniel Pavon, who is botanist in my lab, Vincent Motion, in postdoc for the project, Céline Chatelier, she does her thesis on Teucrium and other plant taxa with taxonomic issue. Frédéric Médaille, Isabelle Lafont-Schwab, who are my colleague in Marseille, professor in Marseille. Olivier Ferreira, Patrice Noforio, the botanist of the National Park. So at Zoology, I start working on the Teucrium because of this man who has some problem to identify the plant in Calanque. Julien Hugo from Conservatoire Botanique Méditerranéen, and the student Stéphanie Tranchat, Anatole Mangendre, Amine Majoub, David Torres, Noé Fleury, Laura Lefebvre, and my colleague from Spain, Juan Viruel, Eduardo Narbona, Montserrat Rista, Nia Roque, Perifraga Ravenbo, and my colleague of Italy, Baqueta and Casaza. And the reason of also of collaboration with Italy and Spain that we start to collect a lot of plants from this country to try to understand the origin of the free population. And there is also some taxonomic issue as this. Okay.
Thank you very much. We have time for questions here and online, not in case.
Bonjour. I didn't really understand the conclusion about your hybrids on your last figure. Then you simulate hybrids, but before you said that you have real hybrids.
Yes.
From a phenotypical point of view in nature, but you don't have their genotype or?
No, I have one. So these plants are diploid, and the hypothesis is that they are from hybrid origin. But as you can see, so this axis hold most of the variants, and they are closer to Oreo, which is the biggest disturbing. So the hybridization. One working hypothesis that was pollution of the south of Marseille was maybe one important factor in this interrogation. But the current result of genetic data shows that this is rather really older than this problem. So I guess that there, phenotype and genetic interrogation tests show that they are hybrid. Now we try to find some more precise morphological variable and also ecological data to see how they are still hybrid or not. And as you see, for the Teucrium ovum, I use Sandbaum genotype, not the genotype from Calanque, because the golden Orion germander is present in this place, but they are very close, in fact, to this. So I think that even the golden germander in Calanque is introduced. So
Yeah, I get it, but I just missed the same.
And in this graph, there are no Teucrium polyum.
no, I do not put it. No, because well, it's not easy to do mixed analysis with diploids and tetraploids. But I have some evidence of hybridization between, for example, this is well, rather common. This one is diploid and this one is tetraploid. But that is a clear ecology. I never saw that on the coastline. They are clearly on the plateau, Ginest, Montpellier.
Thank you. Do you have an idea of the divergence time between the different species? Because I was wondering, like, to what extent you are sure that those are evidence of hybridization and not ancestral population structure? So shared inherited, still shared inherited.
Yes. No, it's a good point. So the genetic differentiation is not very big. So like the maximum FST is 15%. But well, I start to do some coalescent analysis, and we'll quiz. Most of the effort of the last year was trying to connect, but it's fine. The answer to your question is to try to connect this population to a wider phylogenetic context at a biogeographical level, and so we spent a lot of time to collect and to analyze, and well, we'll see if we can at least estimate this knowledge. And also the contact, if it is really an hybridization, the contact between maybe the golden germander, the ancestor, and this one. And because this is an Iceland, but 8,000 years ago that was connected. So possible to connect the geological and geographical context.
I had one question that you mentioned a bit on pollution. Could you develop a bit on the hypothesis of pollution behind?
Oh yes, there is two hypotheses, two different kinds. So this place is really a nightmare of pollution. Currently, the ADEME start to do the depollution and try to, and the soil have it's very heterogeneous, but you can find some very high concentration of chlorine, arsenic, all this kind. And so one first hypothesis that maybe there was a relationship between genetic recombination, adaptive integration to the new stress caused by the pollution. A second, so a direct hypothesis of selection. This is known in some other cases. So like the methodical hypothesis. A second kind of hypothesis is an environmental. One is that if you observe an old picture of this place that was clearly totally open, no tree, no vegetation, no high vegetation. So that was at the moment a paradise for germanders that are adapted to stress. So one hypothesis that maybe during the last century there was a clear environmental connection between the coastline and the summit. that could have helped to hybridization. And so this is an indirect effect of the industry that cut all the vegetation because they pollute a lot the place. And,
But you said, given that you see the PCA that they are closer, you now have a new study that was earlier the pollution separation.
yes.
So
Yes, but anyway, it's true that when we collect and observe the maximum of diversity, that was another place that are at few meters of the pollution. One difficulty in this project is these plants are very long to cultivate. So we have some young plants in the garden, but still not flowering. And they will flower at the end of the project. Their phenotype.
they live a lot.
Yes, they are perennial plants.
Do you have an idea of the longest life of this plant? No? 10 years, 20 years, 30 years?
Maybe more because at least, for example, this one, it's well, even this one. They die. The part of the upper part die each year, and they rose through.
Yes.
So can be very old. No, it can be very old. Maybe we have a young colleague in Marseille now, and he's working on dendrochronology on air plants. So maybe we'll observe this. But one interesting point is that when we look at the genetic diversity on the Frioul Iceland, it's important. So the effective number is maybe close to 1,000 of individual, and this is interesting because it's not a big population, but and I think there is no evidence from the genetic data of genetic integration in the Frioul Iceland. It is really isolated. So it's not the case for the Rio.
Pollinated by which kind of insect?
What?
What?
No, there is a thesis on this. A lot of hymenoptères, but also coléoptères. Il y a une soutenance de thèse là-dessus.
If I come back to your introduction about uncertain species, in the second example, I mean, you have two genetic clusters very strong with no intermediate genotypes that are in contact, even maybe in sympathy. And so the problem is not the species, the problem is the phenotype. And if genetic markers were cheap, and if they were routinely used, if they were operational on the field with the ants, actually, that is great. But if they were, you have no problem of species. That means you have no hybrids, they are in contact.
No, well.
The problem is the link between how to phrase it as a question. Is the problem the link with the phenotype more than the as much as your perfect example? Clearly, you are in the gray zone, but here, are you really in the gray zone?
Yeah, yes. Well, it's not a problem, a species issue from an evolutionary point of view, but from a taxonomy Yes. So probably we try to change just the taxonomy and restrict the name presence to the frame element. But then all this population will lost their protection. And we are in the National Park of Canada, but the problem of the National Park of Canada is that the city is entering the national. In some city in Europe, sometimes the national park are going into the city. Here, this is the contrary. The city is going into the mini. Here we are in Marseille, and there is each year there is problem of voilà. Enedis change les poteaux électriques, font des trous partout, etc. Enfin, and now there is a people. So if we change the taxonomy, this one will lose its protection statute. So what I look now is if I have some argument from a scientific point of view, design this not as an hybrid zone, but as something of hybrid origin. So at the beginning of the project, when we look at the phenotype, we design this as an hybrid zone. And you know that in plant, hybrid zones are not so easy to analyze and to describe. They are not well spatially structured. And now, after the observation that the most distant, the most differentiated cluster was this one, well, it's not an hybrid zone. So what is it? This?
It's what I was saying. It's not a hybrid.
No, yeah.
The point is that to recognize a cluster, you need genetic markers.
Well, so we are looking now to more precisely to small character to maybe find a combination of character on yes. So that could be subspecies of Orion. I don't know. So there is an idea to also to clearly use taxonomy to because well in conservation and even at a small level, the main problem is that populations are not recognized as for protection. So, and this is not a common plant, and I mean this is also. Other question online.
Okay, thank you very much.
Okay.
De la classe, excusez-moi, travailler.
Par exemple.